Okay, we can start now. Welcome to the fourth session of virtual seminar series on hemoglobinopathies. Today we have renowned clinician scientist, Professor Mark Walters from University of California, San Francisco, UCSF, who is going to deliver a lecture on curative therapies for hemoglobin disorders. Thank you so much, Professor Walters, for accepting our invitation. And it is a great honor and privilege for us to have you at our seminar series. Today's session will be chaired by Dr. Rahul Bhargava. Dr. Bhargava is a leading hemato-oncologist and director of hematology and bone marrow transplantation center at Fortis Memorial Research Institute, FMRI, Gurgaon. This is near to Delhi. So now I hand over the session to Dr. Bhargava. Dr. Bhargava, please.
Thank you, Shiva. It has been a great pleasure to be chairing this session. And it is an immense pleasure to introduce Professor Mark Walters. He is a Jordan Family Director of Bone Marrow Transplant at UCSF, Benioff Children's Hospital, Oakland. He has earned his degree from University of California, San Diego School of Medicine. He's completed a residency in Pediatrics at University of Washington School of Medicine, followed by fellowship in pediatric hemato-oncology at the Fred Hutchinson Cancer Research Center, which is one of the most renowned centers in the world. Dr. Walter is a national scientific, clinical, and education leader in hematology, has published many peer reviews, and has received NIH grant awards to support his function works. With NIH and industry support, he has been conducting a number of multi-center clinical trials for sickle cell disease and thalassemia. And of course, in Afro-African population, sickle cell is a huge problem. And even the president of the United States stays committed for finding a cure for sickle cell. Dr. Walter has worked with industry sponsors and taken a lead investigator role in early phase clinical trials for gene therapy for thalassemia major and SCD, which is a Bluebird biocon, which is currently in news these days. Currently is focused on initiating early phase clinical trials to test novel genomic editing techniques in hematopoietic stem cells for individuals affected by hemoglobin disorders. And he is also the clinical lead investigator in planned trial to inactivate BCL11A in hematopoietic stem cells to reactivate fetal hemoglobin in subject with thalassemia, which Shiv also is doing right now with CRISPR technology. So, Professor, it gives an immense pleasure to introduce to Indian audience and across the world. The idea is that all of us are looking for some kind of a cure. As we know, India is the third largest country in the world having a sickle cell population, with around 53 districts being affected, and of course give rise to 10,000 babies every year for thalassemia major. Though the transplant has come up with its own curative strategies, but it's still far from reality that every person can get a transplant, though the haplotransplant is picking up in thalassemias and sickle cell. So we are one of the largest transplant centers in the country doing sickle cell transplants from Africa, mainly from Nigeria, Zimbabwe, Uganda, Zambia, and so on and so forth. So it will be a pleasure to listen to you for another hour. How and newer strategies are there which can promise us moon in terms of delivering a cure for hemoglobinopathies? Over to Professor Mark Walter.
Thank you, Professor Bhargava, and I also want to thank Shiva for this kind invitation. It's really a pleasure to participate in this series of lectures. I have gotten close to some colleagues in India, in particular Professor Alok Srivastava at the Vellore Christian Medical College, with whom we've struck up collaborations over the years, and also the team at Chandigarh. And I'm really sorry I can't be in New Delhi in person. That would be a lot of fun, but this is the next best thing. So it's really a pleasure to share some of the work we're doing and some ideas we have about the future of curative therapies and hemoglobin, clinically significant hemoglobin disorders. I'd like to begin with this disclosure of my outside activities, none of which I think will have any bearing on this lecture. So to begin, I always focus on the gold standard, which is allogeneic hematopoietic stem cell transplantation for both transfusion-dependent thalassemia and sickle cell disease. And these are two registry data analyses published fairly recently, looking at overall survival after an allogeneic transplantation. And what it shows, first of all, is that when there's a well-matched sibling donor, generally in a child who's in good shape, either with thalassemia or sickle cell disease, the survival and event-free survival exceeds 90%. And in fact, the survival in sickle cell disease these days is more than 95%. Of course, as you all know better than I do, not very many people have a full sibling, a well-matched sibling donor. And you can see the impact that has on survival when an alternate donor is pursued, particularly when using a mismatched relative. So the idea for autologous transplantation using engineered stem cells from a person's own hematopoietic stem progenitor cells is to broaden the availability of this curative therapy. Now, some of the best work, I think, in the world is being done by Suresh Honging and his team at the Ramathapati Hospital in Bangkok, Thailand. And this is an ongoing series of patients he's been treating for some time with transfusion-dependent thalassemia. Now these number 83 patients in total. And he uses this interesting idea of overcoming the problem of the risk of graft rejection and recurrent thalassemia in mismatched donor transplantation by first giving 2 cycles of fludarabine decadron to kind of tamp down the immune regulatory milieu, if you will, and then uses this mild ablative combination of avid ATG, busulfan, and fludarabine. And then, of course, post-transplantation cyclophosphamide to accomplish in vivo T cell depletion. And with this approach, really remarkable, very low rates of severe GVHD with an event-free survival probability of 96%. And I show this slide not because I'm going to dwell on haploidentical transplantation this evening, but just to show that there is this looming alternate. There's a lot of hype around gene therapy, gene editing, but we have this other foundation that I think around the world might certainly be a more feasible way to apply and accommodate curative therapies for these disorders. So what are the barriers to transplant for hemoglobinopathies? Well, first of all, most families will not have an HLA identical sibling donor for an affected child or adult. And if we do an unrelated donor search, because of the fact that most of these disorders occur in underrepresented minority populations, only another 19% will have a well-matched unrelated donor. So even if I wanted to treat everyone who appears in my clinic, I can't. I can only treat about a third. And moreover, clinicians who take care of the patients often won't refer them to me because these procedures carry a risk of graft versus host disease that can be morbid or fatal, and this risk of dying from the transplant itself. So transplant until recently has really been largely restricted to children and applied quite sparingly. So the question we started to ask about a decade ago is, can we modify autologous cells for clinical benefit? And as important, can we ensure equitable access to novel curative therapies once these have been established? So we're well on the answer to number one, but number two is really, as much as anything, a health policy issue with which we're just starting to scratch the surface. But it's as important, if not more important, than the first question. So there are several genomic or gene modification therapies for hemoglobinopathies that are being studied and have been applied. So the first, probably the best studied with the greatest amount of experience, is gene addition therapy. So using a vector of some sort to introduce an anti-cycling beta globin for sickle cell disease, or a fetal globin gene for its anti-cycling activity. And of interest, either one of these, to a certain extent, can also be applied to transfusion-dependent thalassemia. And then there's gene editing of the regulatory genes involved with fetal hemoglobin repression to edit those in such a way as the fetal hemoglobin is increased after the modification. And then work that we're doing with others, gene editing to correct the sickle or the thalassemia mutation. So this is our dream in the collaboration with Alok Srivastava and Vellore, is can we also target the most common thalassemia SNPs in India, perhaps with even a single guide RNA to tackle this problem. And then a topic that I'm not going to cover today, but really is the holy grail to which we'd all like to obtain, is can you do this gene editing in vivo? introducing reagents in such a way, in a targeted way, that they could be delivered to the hematopoietic stem cells in the marrow where they reside to correct these mutations or to elicit the editing changes. So I'm going to start with the Bluebird bio-sponsored trials that I've participated in. These rely on this lentiglobin BV305 vector that's shown here in the schematic. It's a modified beta globin gene. There's a single amino acid substitution, threonine for glutamine. The glutamine has a side chain that sticks out in a way that inhibits and destables the sickle polymer formation. It's also perfectly functional, so it can be used both in thalassemia and in sickle cell disease. And the expression of the gene is under control of beta globin regulatory elements. So this cassette should only be expressed in the lentiviral vector in erythroid cells. And in fact, this is a self-inactivating replication deficient lentiviral construct, so it can get in, but it can't excise itself once it's integrated into the genome. So this single amino acid substitution allows one to monitor the hemoglobin level in the blood post-infusion because it can be detected by HPLC reliably. And I just mentioned that it acts much like the gamma, the fetal hemoglobin, in that the T87Q hemoglobin molecule will interfere with hemoglobin S proliferation. And the idea is that if one could attain a level of 30% F plus this modified hemoglobin, it would have disease-modifying anti-sickling activity. So this is how the therapy works. I think most of you are familiar with this by now. So patients are referred to us. They undergo mobilization in thalassemia with a combination of GCSF and plerixafor. In sickle cell disease, just a single agent plerixafor. The cells are collected by apheresis. They're then shipped to a manufacturing facility where they undergo CD34 positive selection. They're transduced with the lentiviral vector. And if they meet criteria for release, they're cryopreserved and then ultimately sent back to us at the transplant center, frozen, where we administer single agent busulfan for myeloablation and then thaw the cells at the bedside, reinfuse. And they're in the study for 2 years with the option of participating in a long-term follow-up study that extends through 15 years post-infusion. And this is the talented team here in the inset that's done all the heavy lifting to get the logistics of patients mobilized, collected, and then treated. So we published this experience in transfusion-dependent thalassemia now, gosh, nearly 3 years ago. This was the North Star HGB 204 study, lentiglobin gene therapy. And its primary objective was to establish the safety and efficacy of lentiglobin gene therapy in thalassemia. And all 18 patients have now completed that initial 2-year study. Most of them have, actually all of them have enrolled in the long-term follow-up study, and there's now a median follow-up of nearly 3 years. So we think about these disorders according to their genotypes vis-a-vis how they respond to gene therapy. So the non-beta 0, beta 0 genotypes, this would include, for example, E beta 0 thalassemia. So these are genotypes that express some hemoglobin endogenously, but not sufficient for transfusion independence. So they generally start transfusions a bit later in life. Whereas the beta 0 genotypes, these are more severe. They make no endogenous hemoglobin, and of course start transfusions in the first 6 months of life. And these swim lanes, that's what we call these, they show the time before transfusion independence was established in light blue, in months after the drug product was infused, and then in dark blue, the time since their last transfusion was administered. In some cases, nearly 5 years since their last transfusion. And 4 of the 8 patients with the more severe genotype transitioned to transfusion independence, compared with 90% of those with a non-beta 0, beta 0 genotype. And their hemoglobins at last visit are shown. They range quite widely, 8.3 to 12.2 in the group in the upper panel, and around 9 to 10 in the lower panel. So, but in all cases except for one, the idea was to have a hemoglobin of at least 9 to 10 independent of transfusions. So the phase 3 studies are ongoing, North Star 2 and 3, and 2 enrolled patients just with the non-beta 0 genotype, and North Star 3 enrolled patients with the beta 0 or the with the IVS 1110 allele qualifying as a well, it's a severe beta plus, but in this study it was called the beta 0 because that's how it behaves clinically. So median follow-up of 14 to 19 months, and now all 41 of these patients have had the drug product infused. And what I thought I would do today, since I'm a pediatrician, just focus on the outcome in the patients less than 18 years of age. So their characteristics are depicted here. So there are 13 patients less than 12 years of age, 11 adolescents between 11 and 18 years of age. Almost none of them had splenectomy, as you might imagine, and the time at their first transfusion varies, some quite late and others within the first 6 months of life. Most of them had elevated liver iron concentrations at baseline, not all of them, and there was no significant iron loading at the baseline. So most of them were mobilized with a single cycle of GCSF, plerixafor, and apheresis. 2 patients had to have 2 cycles. The vector copy number of interest was a bit higher in the adolescents compared to the younger patients, and that translated in having a median of 82% of the CD34 positive cells transduced compared to 70% in that younger group. Although the cell dose in terms of CD34 positive cells per kilo was pretty similar, in all cases met the target of 5 million CD34 positive cells per kilogram recipient weight. All of them received myeloablative busulfan, but interestingly, many of them, because these are children, I suppose, received Q6-hour dosing rather than daily dosing of the busulfan. And this shows the time to hematopoietic reconstitution. There was no difference in the 2 groups, really. 26 days to neutrophil recovery, ANC greater than 500, and 50 days for platelet recovery with a transfusion-free interval of at least a week and a platelet count over 20,000. The time to lymphocyte recovery is shown on the bottom panels, and the area highlighted in gray is the normal range. And this shows that it took anywhere from 6 months to 12 months, and in some cases longer, for the T cell, B cell, and NK cell populations to recover numerically to the normal range. So a bit longer. And so generally we treat these patients as being immunodeficient through at least 6 months post-infusion, and then start the process of re-immunizing them, and also relaxing some of the infection control procedures. The safety is as you might expect after myeloablative busulfan. So the most common non-hematopoietic grade 3 or higher adverse event was neutropenia, febrile neutropenia, or mucositis, or reduced appetite, some bleeding. Serious adverse events are listed, again, mostly fever or febrile neutropenia, mucositis, and then 2 cases of veno-occlusive disease. So in total, even those who were children and they should have been in better shape than the older adult patients, still had three significant episodes of veno-occlusive disease that was treated with defibrotide. And 2 of these were serious grade 4 events, patients 12 years of age. So just underscores how tricky busulfan is in the thalassemia population. There was also a problem with thrombocytopenia. Some of them had delayed platelet recovery. And so there was 1 non-serious grade 3 thrombocytopenia that was scored. But otherwise, there was no graft failure. None of the patients received their unmodified rescue stem cell. And then all of the patients are alive. And finally, no replication competent lentivirus, or importantly, no evidence of insertional oncogenesis. So this is screened by looking at integration sites serially over time post-infusion, and the trigger is that if any single insertional site representing a clone of hematopoietic cells. If it exceeds 30% of all of the insertion sites, then that triggers a workup for clonal hematopoiesis and myelodysplastic syndrome. So none of the thalassemia patients have, the children or the adults for that matter, have fallen into that adverse event area. And then this shows the transfusion independence in the two groups. So the older adolescent group in the upper panel, the younger patients less than 12 years of age in the lower panel. And the total hemoglobin at the last visit is shown. And so some of these are, particularly in the younger patients who did not transition to transfusion independence, are low. And in fact, the weighted average was 11.4 in the older patients and 10.1, so lower in the very youngest patients, with a duration of follow-up that was about 15 months. So 4 of the 6 patients less than 12 years of age achieved transfusion independence compared to all 9 patients who were 12 to 18 years of age. And then this is another way of depicting the data. So the pink bar is the hemoglobin AT87Q, so that's the gene therapy hemoglobin. And then the endogenous hemoglobins are shown. So these are all at month 24, month 18, et cetera, month 12. These are long after the transfusions have been stopped. So any hemoglobin A that's detected at these time points is endogenous. And then the green is there's one patient who had hemoglobin E, beta 0 thalassemia. So to my eye, the pink bars are a little bit lower perhaps in the younger group compared to the older group, but overall in a similar range. And then, so what happens to ineffective erythropoiesis when this treatment improves the hemoglobin but doesn't correct it completely? So in red are the youngest patients, less than 12, and blue are the patients between 12 and 18 years of age. So the normal here for soluble transferrin receptor and absolute reticulocyte count is again the gray area. And again, I don't really see an obvious effect of age. Perhaps the soluble transferrin receptor stays in a higher range in the younger patients compared to the older patients, but that would be difficult to say. In most cases, though, there's this trend towards normalization, which is very good. But the MDE ratio, the myeloid erythroid ratio in the marrow, improves, but really doesn't correct to the 3-to-1 classical ratio. And then there are photomicrographs on the lower right-hand corner showing at screening the proliferative marrow that you expect in transfusion-dependent thalassemia, and then after treatment, improvement in the in the cellularity, even some hypocellularity, perhaps, in the examples shown in this slide. So I'm going to shift now to sickle cell disease. Again, this is using the same lentiglobin vector, and this is the HGB-206 trial. So this is going to focus on the third group of enrollments called Group C that targeted patients between 12 and 50 years of age who had a history of severe vaso-occlusive episodes. So they had to have 2 or more for 2 consecutive years before enrollment that was either resistant or to hydroxyurea or patients who are intolerant of taking it regularly. So enrollment in this study also has been completed and in fact suspended. We'll get to that. And the outcome was for achieving the key outcomes were having a weighted average hemoglobin AT-SMQ that accounted for 30% or more of the total hemoglobin for at least 6 months or longer post drug product infusion, raising the hemoglobin by 3 grams or having a total hemoglobin 10 grams or higher, again, for 6 months or longer post infusion. And then the clinical outcomes are to have a 75% reduction or more in severe vaso-occlusive episodes in the 24 months post infusion, and then looking at the proportion of patients who had complete resolution of these severe vaso-occlusive episodes. So, oops, sorry. So here's the patient characteristics. 43 were enrolled, age consent, median 24. The span of ages is shown. And 39 had severe vaso-occlusive episodes with a median annualized rate of 3.5 per year. And then acute chest syndrome, priapism, and any stroke accounted for a smaller proportion. So this really focused on pain as the primary eligibility criterion. Whoops. And then, so unlike the thalassemia patients, these are sickle cell disease patients. We can't use GCSF because it can precipitate acute chest syndrome and other vaso-occlusive complications. So this is plerixafor alone, and this took a median of 2, and in one case as many as 4 mobilization cycles to collect enough CD34 positive cells. So although the collections were really quite good in many of the patients, median of 10 million cells per kilo per cycle, they all received myeloablative busulfan. The time to neutrophil engraftment and platelet engraftment is shown. They were in the hospital for about 5 weeks. The vector copy number on average was 3.8. This translated in having 80%, median of 80% of the cells transduced. And the cell dose ranged from 3 to 24 million per kilogram with a median of 6.8. So this is perhaps the most important result of the study. These are more swim lanes. So these are the patients in the 24 months before they were enrolled in blue, and then in the 24 months after they were enrolled. There's something wrong on the bottom panel. So in yellow. So the red diamonds are any vaso-occlusive event. I'm sorry, severe vaso-occlusive events, generally leading to hospitalization. The blue are any sort of vaso-occlusive event. And there's lots of them in the 2 years before they gave informed consent. And then after infusion, they just essentially go away. So this represents something like a 99.4 reduction in the severe vaso-occlusive event incidence. So that's really amazing and probably the most important finding of the study. Now there are other important findings. This is one of them. This is a patient reported outcome instrument called PROMIS 57, and this is just looking at pain intensity. There are 2 colored curves here. The green one are patients who had a baseline above worse than the norms in the population, so a score of 6, and then with no pain being 0 and terrible pain being 10. And then there was a smaller group of patients who at baseline actually were in the normative range. And in both groups, there were improvements in the pain score over time through 12 months post-infusion. So, and there's more information about this that'll be reported this summer at EHA, but this is the direction of these domains that are reported by patients using the PROMIS 57 instrument. Then here's the equivalent of what I showed for thalassemia, and the green here is hemoglobin S, and the red is the T87Q, and then the hemoglobin A, which is transfused. You can see at the 3-month post-infusion time point that goes away, and by 30 months post-infusion, there's a very nice balance of about 50% T87Q and 45 to 50% hemoglobin S. And then the total hemoglobin settles at a median of 11.5 grams per deciliter. So it seems to satisfy all those endpoints that were targeted at enrollment. And so this looks like sickle cell trait, but that would only be true if in fact each red blood cell had a mixture of hemoglobin S and hemoglobin A, or T87Q. If in fact this is the same as giving transfusions, this would mean that there could be 46% of the cells at 30 months having 100% S and 52% of the cells having 100% hemoglobin A T87Q. In that setting, you might expect there would still be clinical events because half of your cells in circulation are still prone to intravascular cycling. So to get at whether this was pancellular or heterocellular distribution, the Bluebird bioscientists did the following assay, and they made antibodies for both for hemoglobin S, hemoglobin A, and then hemoglobin A T87Q. So using the antibody, this is a single red blood cell Western blot. It's not quantitative, but it's qualitative. Using the antibody for S, for example, if there were just S cells in the circulation, they would stain in this schematic orange. But if, I'm sorry, that was hemoglobin A. If they had S, they would stain just blue. If, and then the depiction I just explained, if you have this mixture of cells that have all S or all A, you would see this combination of collection of orange and blue cells according to staining. But if there's, and these would, as I said, would be prone to intravascular sickling. But if there's this stippled pattern, like you see in sickle cell trait, all of the cells would have fairly equal quantities, well, somewhat more A than S because the S is unstable, then that would be consistent with S trait. So the question was asked then, is this what the red cells look like after the Liposite gene therapy? So there are still some cells that have 100% S, but the pattern isn't more stippled, showing a mixture of both S and T87Q hemoglobin in the same red blood cell. And in fact, the stippled pattern is what was observed. So in this timeline from infusion, so preconditioning, these patients are transfused. So this shows exactly what you would expect. So a small fraction of the cells still contain 100% S, and the rest have hemoglobin A from transfusions. And as the A cells go away when transfusions are stopped by 6 months, most of the cells are stippled, and at 24 months that pattern is sustained. So 90% of the cells have both S and hemoglobin A27Q in them, and there's a small remaining population of red blood cells in circulation that just have hemoglobin S in them. So, and in fact, when, so this is non-quantitative, but when quantitation of the level of the hemoglobin A27Q per red cell was measured, it was 15.3 per cell. And that compares with the 13 to 18 picograms per cell you would see in individuals with sickle cell trait, and this benchmark of 10 picograms of F per red cell in patients who have hereditary persistence fetal hemoglobin. So a very favorable range that would suggest that this should be having a strong clinical effect, just as we observed. And in fact, the hemolysis, markers of hemolysis also begin to normalize. So the reticulocyte count, the lactate dehydrogenase total bilirubin, over time begin to migrate into that normal range highlighted in gray. So what about adverse events on the study? So again, most of these that were grade 3 or higher have to do with receiving mild beta-busulfan, so febrile neutropenia, mucositis, liver function test abnormalities, and then more serious adverse events related to abdominal pain, nausea, some opioid withdrawal syndrome, vomiting. So of interest, unlike the thalassemia cohort, no cases of veno-occlusive disease of the liver, and only one patient with a non-serious grade 2 drug product related adverse event. No vector-mediated replication competent lentivirus, and no, until recently, insertional oncogenesis. That's still the case, but this is now under investigation. There was a young patient, 27-year-old, who died at home suddenly 18 months after infusion of the drug product. He had been doing very well, but had a significant baseline history of cardiopulmonary disease related to sickle cell. And in fact, his autopsy showed cardiac biventricular dilatation. So he seemed to have cardiac interstitial fibrosis, and that might have been the cause of sudden death. There was no pulmonary embolism or stroke. And so this, to me, underscored the importance of taking patients to this treatment before they have these kinds of problems that could predispose to sudden death. Now, I'm sure you all have heard in the headlines over the past 2 weeks that Bluebird Bio announced a temporary suspension of these studies, both the phase 1/2 and phase 3 studies of lentiglobin gene therapy for sickle disease. They also suspended using the BB305 vector in Europe, the Zynteglo that's been licensed by EMA in Europe in thalassemia. And the reason it had to do with a case of AML that developed 5 years post-infusion in one patient, and then a second suspected unexpected serious adverse event of myelodysplastic syndrome in a patient who was about 6 months post-infusion. So this is under investigation. And then of interest, the National Heart, Lung, Blood Institute that has been supporting a phase 2/3 trial of lentiviral gene therapy to inactivate the BCL11A target and induce fetal hemoglobin by investigators at Boston Children's trial also has been suspended pending the outcome of this evaluation by Bluebird Bio. And specifically, the question is this related to the busulfan alkylating agent administration in recipients with sickle cell disease who are already predisposed, perhaps, to develop acute myelogenous leukemia because of the disordered marrow that's a characteristic of this disease, or is there evidence that there's insertional site oncogenesis, which would, of course, completely change the thinking about the safety of this vector. So this is now the third case out of 40 or 45 patients who've been treated with sickle cell disease. And these are data from a paper that was published last year that provided some evidence suggesting that it was the busulfan and not the vector that caused this patient's myelodysplastic syndrome with monosomy 7 and AML. This is a patient who ultimately died of the AML after an unsuccessful haploidentical transplant. And so this is the integration site analysis. The vector copy number in this individual was quite low, 0.1 to 0.2 post-infusion in the peripheral blood. And the colors indicate unique clones with a single integration site. And the 30% mark is right about here, then that would ordinarily trigger investigation if any single clone like this one in red reached the 30% mark. So it didn't in this particular patient, although it came close. But of interest, I know you can't see this very easily, but the top most common integration site resembled over time changes at every 6-month interval. So these things, because the stem cell firing stochastic, no one single integration site clone ever dominated over time. And then this just shows in the bottom using a staining for glycophorin. That's the brown staining. These are erythroid cells, and then these red dots are probes for the lentiviral insertion. And so what they were able to show is that none of the leukemic blasts had, at least by this staining, evidence of an insertion. And then this is again shown in this slide. So when they selected for CD34 positive cells that were myeloid leukemia cells, the vector copy number was really below the level of quantitation, 0.02, compared with 0.14 in the unsorted bone marrow sample. So there really wasn't any detectable lentiviral vector in the leukemic population. And the investigators, and I was a co-author of the paper, we all agreed that this was very unlikely to be a case of insertional mutagenesis. So whether or not that's responsible for the two latest cases, hopefully we'll have some answers in the next month or two. So perhaps because of that, or in addition to that, there's a growing interest in using the CRISPR-Cas9 reagents to accomplish the same curative outcome. So this is from the New England Journal paper that was published in print last month. It's been out a couple months ago. This is from the CRISPR Therapeutics trial, targeting BCL11A. So this is the master regulator of fetal hemoglobin. It's sufficient and necessary for repressing fetal hemoglobin at birth. So this is birth, and this is where we have this transition from gamma globin expression that's turned off and beta globin expression that's turned on. And what this team has done is engineered a guide RNA that targets a GATA1 binding site in the erythroid-specific intronic enhancer of BCL11A. This is all shown schematically. Creates a double-stranded break that when it's repaired, the GATA1 binding site is disrupted, and then the enhancer is inactivated. And with an inactivation of this erythroid-specific enhancer, BCL11A is knocked down or knocked out only in red cells, but not in the other lineages. And as its name implies, it's absolutely needed for not just B cell development, but also hematopoietic stem cell proliferation and maintenance. So this is another schematic, and this just gives credit to Vijay Sankaran and Stu Orkin, who discovered this, and they proved this nicely in a conditional knockout in a sickle cell disease mouse. I believe this was the Towns mouse. And so the idea is that if you repress BCL11A, this allows de-repression of the gamma globin expression, and in a sickle cell mouse should make it better. And so that's exactly what happened. Using this conditional knockout, they were able to transition this very abnormal sickle cell smear in the sickle cell disease mouse to a normal smear after the BCL11A was repressed. So this is the clinical trial. They use a drug product called CTX001. They introduced the CRISPR-Cas9 guide RNA RNP ribonucleotide protein as a cassette that's electroporated into stem cells. It targets the erythroid enhancer, and then post editing, should de-repress the gamma globin genes and allow fetal hemoglobin to be expressed at a high level. And this is what they reported. These are two patients. The one on the left, transfusion-dependent thalassemia. The one on the right, sickle cell disease, and really spectacular result. Hemoglobin on the panel on the left, 14 grams per deciliter, almost all of which is fetal hemoglobin in this patient with a severe form of beta thalassemia, and transfusion independent. And then in sickle cell disease, a similar pattern to what we saw in the Bluebird bio-sponsored trial, but in this case, the S in purple is balanced by the fetal hemoglobin in blue, and this sort of similar ratio. As it turns out, also pancellular distribution of the fetal hemoglobin. And this is what happened clinically. So this is transfusion events in the panel on the left before screening. Each circle indicates a transfusion encounter, and then within 30 days post-infusion, the transfusions were stopped, and there's now 21.5 months of follow-up where this patient is transfusion-free. And then in the on the panel on the right, not shown here, are both vaso-occlusive events. These are, I think, all hospitalizations and transfusions with cessation of both within about 2 months post-infusion. So no pain and no need for ongoing transfusions in this sickle cell disease patient. So really quite an interesting and spectacular result for these first patients presented. Now, in the same journal edition was a publication of this study led by Dave Williams, the PI. The lead is Erica Ezrick, who's an investigator at Boston Children's Hospital, and they're using a different technique to knock down or knock out BCL11A. And so this is a lentiviral vector with those same erythroid regulatory elements, with the beta globin promoter driving expression of a microRNA derived scaffold. So what this RNA does is it, when it's expressed, it targets a region in the BCL11A messenger RNA and inactivates it. And this mimics other endogenous microRNAs. It leads to more physiologic, lower expression levels, less non-specific toxicities, and allows for regulated erythroid expression and avoids expression in B cells and hematopoietic stem cells that I already mentioned is problematic. So same approach, they mobilize their patients with sickle cell disease with plerixafor only. They also receive myeloablative conditioning with busulfan, and then infusion of these gene-modified cells with 2 years follow-up. And the results were equally striking. So the graph on the left, these are 7 patients who've been infused, shows the percent fetal hemoglobin over time in months post-infusion, settles out at around 25 to 30%, which is exactly what you would hope to achieve for a strong clinical effect. And then on the right, the F cells. So these are mostly now 70 to 80% of the red cells in circulation are F cells or fetal cells, so have a very high fetal hemoglobin level and should be associated with a strong clinical benefit. And in fact, here are the hemoglobins in these 7 patients ranging from 9.5 to 11.4, transfusion independent. Here's the fetal hemoglobin fraction in the 20 to 40% range. And then here again, we're trying to target 10 picograms fetal hemoglobin per F cell, and in most cases that is getting achieved. So this too was associated with a strong clinical effect with regard to improvements in hemolysis and clinical complications. And this is the phase 2 trial that's now being supported by the National Heart and Lung Institute and is on hold until we understand more about the genotoxicity risk and other risks for that matter of this approach in sickle cell disease. So I'd like to finish with some work that we're doing in the laboratory, and this has really been a remarkable team. Mark DeWitt, who is at UCLA, was project manager after being a postdoc in Jacob Corum's lab at UC Berkeley. When Jacob left, Mark moved to UCLA and continues to lead the operation. And then important contributions by David Martin, Wendy Magus. Dario Buffelli is one of our bioinformatic experts with Stacia Wyman at the Innovative Genomics Institute, Berkeley. And then Don Cohn, co-leading with Fyodor and me the whole project. And he heads the CMC manufacturing operation while I'm leading the clinical translation and hopefully we think a clinical trial soon. So unlike the CRISPR-Cas9 targeting of BCL11A, which is basically creating a double-stranded break followed by insertion deletion at the editing site, what we have to do to correct the sickle allele is something we term homology-directed repair. So we're actually making a double-stranded break near the sickle mutation, and then using replication and extending replication fork and an oligonucleotide template that has the normal sequence repairing the sickle allele. Now, you don't get anywhere near 100% correction rate in stem cells using this technique, at least in our hands with the current technology. But as it turns out, you don't really have to. And what we've observed over the years after allogeneic transplantation, that post-transplant chimerism with as little as 20% of the patient's marrow made up of donor cells is associated with cure both for sickle cell disease and thalassemia. So what happens is there's this natural enrichment of the healthy donor red cells, such that because of their longer lifespan in the circulation and because they're not prone to ineffective erythropoiesis, you actually get to this natural enrichment of the donor cells such that they're cured. So they don't look any different from those that have 100% engraftment of donor cells. So our target was to correct at least one sickle mutation and at least 20% of the engrafted stem cells. And if we could do that, we think this could be curative. So here's the evidence that, in fact, we're able to do that. And this is the oligonucleotide depicted, and then the guide RNA target. And so this is where the cleavage begins, and here's the sickle cell mutation. So if the cleavage happens here and the oligonucleotide is the template, the replication falls in this direction, and then it is until it gets to this point that the T to A mutation that's responsible for the valine 4 glutamine swap occurs. So if the replication fork falls off before it gets to the sickle mutation, there's still some HDR, and you know that because you also get rid of the PAM site because you don't want to re-edit this site after you've done it the first time. So those can actually be controls for expression in the xenografts that we tested this in. So this is what it looks like schematically. And then taking hematopoietic CD34 positive cells from donors with sickle cell disease, we were able to accomplish this in the mice. So what we do is we infuse the mice with the human cells. We harvest them 16 weeks later, and then collect the bone marrows and make colonies. So each colony represents a single cell clone. And we genotype those single cell clones and ask, is that colony still harboring uncorrected sickle mutations? Is it harboring at least one corrected allele? Or have we created an indel, so an insertion deletions such that HDR didn't occur, and we've created now a thalassemia allele. So two of these, the red and the green are bad, and the blue is good. And the different shades just have to do with whether it's heterozygous or homozygous. And so again, our target is to have, say, 40% or more of the cells. And in fact, in these two, both clinical scale run and sickle cell disease donor run, we've accomplished that, we think, by this colony assay. So is this really sufficient for clinical effect? Or are these indels, so the creation of a new thalassemia allele, is that going to be problematic? Well, we think not, and this is really the crux of the matter in the next slide. So this is, again, each of these represents one mouse of 80 or so that we xenografted, and it looks at populations from the marrows in these mice based on their being unfractionated, CD34 positive selected, or CD235A positive selected. So these are glycophorin expressing erythroid progenitors. So stem cells, erythroid progenitors, if you will, and then unfractionated marrow. So this is the unedited control, and the fraction of these allele of the sickle or corrected alleles is not different. And then this is the control I mentioned where the HDR started, corrected the PAM site, but didn't make it to the sickle allele. And again, there's no difference in representation of these populations in the mice. But look what happens when the HDR occurs and the sickle allele is corrected. Look at the fraction of alleles in the erythroid compared to the stem cell populations. Well, they're markedly enriched. In fact, they account for 50% of these type of cells in the marrows of this mice. So this seems to prove to our eye that there is a natural enrichment occurring, and if we had this sort of fraction, that might be curative. For the non-homology end joining, these are the insertion deletions that create an out of frame allele. They drop out. So just as you would expect, a thalassemia allele shouldn't contribute to the red cell population. In fact, they don't. And interestingly, there are some non-homology end joining insertion deletion events where it's in a fraction of 3, so it's in frame, and they also will persist, and perhaps a little bit better than in the stem cell and unfractionated marrow components, but that's probably not going to be significant. But this is significant, and we made a strong enough case that the FDA agreed to grant an investigational new device approval so that we can, with funding that we're trying to procure, go on to conduct an early phase clinical trial with this technique. So I'm going to finish with the ongoing challenges of gene therapy for hemoglobin disorders. There's still a lot of concern about myeloablation and its associated toxicity, in particular in sickle cell disease, whether that ablation causes an increased risk of leukemia, which it certainly might. Limited long-term transgene expression data. There was a patient with thalassemia major treated with an early version of that BB305 vector who, after 10 years of being transfusion-free, did resume transfusion. So is there silencing? Hopefully not, but that's still an open question. Is there a risk of clonal hematopoiesis or selection of proliferative clones? And there's really limited long-term outcome safety, and this is going to be terribly important. So we're all working out ways to screen and then follow clonal hematopoiesis if it develops post-infusion. But that's really terribly important, obviously, particularly in the children who are going to carry these modified stem cells for the rest of their lives. And then, will the cost and complexity of the treatment restrict access to care? Are we really talking about broad versus limited availability, with availability really driven by healthcare resources, which is not really where we want to end up. So this is my last slide. The ideal cell therapy profile, I think, would provide protection from hemoglobinopathy-related complications, both clinical and subclinical. There should be acceptable toxicity profile in the short-term and long-term. Should be accessible and available to most patients, safe in children and adults. And then ideally, we would perform a clinical trial that compares this curative therapy to supportive care and shows a benefit of the curative therapy in the short and long term. So this is really pie in the sky, but it is hopefully we're all working toward in India, in the United States, in Africa, really South America, around the world, Europe. So thanks very much. I want to acknowledge the sites and the Bluebird Bio team for the HGB 204 and 207 gene therapy trials for thalassemia. This lists the sickle cell disease investigators, both from Bluebird Bio and around the international sites. And this acknowledges the funding that I have to conduct the studies that we've participated in. So thanks very much for your attention. Hopefully we'll have some time for some questions.
Thank you, Professor. It was a wonderful listening to you. So there are plenty of questions. You have thought-provoking thing which you have told us about. And thanks for mentioning India. The CMC Vellore is leading the thing. Up north in India, most welcome certain times, because we have got a huge load of sickle cell as well as thalassemia, as well as a African variant, Middle East variant, and Indian variant. So all three variants, if you want to go through, where the HbF level is 3% versus Indian variant, where the HbF level is 30% or 20%, and still there's a disease. It's a very interesting phenomenon of a phenotype and a genotype discrepancy in sickle cell, as we see from Nigeria to Iraq to India.
Yeah, so do you think those individuals should be excluded from a fetal hemoglobin inducing therapy?
So in India, that's what the problem is going to be, Dr. Professor. Generally in India variant, across India also, the HbF varies quite a variable process from 10% to 20%. How further increasing it is going to affect the phenotype of the disease will be a very interesting phenomena to see.
Yes, I agree. I'd like to hear how others are approaching that, because that's really fascinating.
Yeah. So coming to the questions, before I'll ask my questions, there's a from Sangam Giri Goswami. For a sickle cell disease, which therapeutic strategy is better and safer? HBB mutation correction or anti-sickling gene introduction or fetal hemoglobin induction, and why? So just to give a glimpse is why, because the disease heterogeneity is so much in sickle cell. Is it the gene editing or repressing the gene, BCL11, what you have told us, or lentivirus strategy? Because all the phenotypes will be different. How do you approach that?
Yeah, well, that's gosh, if I knew the answer to that, I would just do it, right? So I don't know the answer. First of all, we haven't done any HBB mutation correction in patients, so that whether that's has any efficacy and its safety profile is completely unknown. So that isn't really on the table yet. The gene addition and the fetal hemoglobin induction strategies look quite promising with regard to efficacy. And I guess what they all have in common is this exposure to busulfan. So really what we need to know is there any insertional oncogenesis occurring that would lower your enthusiasm for that approach compared to, say, a CRISPR targeted BCL11A knockout? Or on the other hand, if it's just busulfan, then they all will potentially have the same concern. Or is it the ex vivo manipulations that both procedures require? So you collect the cells, you incubate them for up to 60 hours ex vivo to get them to expand so that they can be transduced or so that they can be edited. And then is that really the problem? And then question has to be which one is worse? And I don't know the answer to that. And finally, if you follow the gene editing scientific literature, there's a suggestion that the correction of the double-stranded break is, and the tolerance of that is higher in TP53 deficient hematopoietic stem cells. So are we then enriching for TP53 deficient stem cells that we're putting back into patients? And because this therapy is so new, maybe we'll begin in 10 years, we'll start to see problems related to that. So I just don't have the answer until the toxicity questions have been determined. They all, though, appear to have efficacy favorability, and that's what's, I think, exciting. So So we're going to spend the next 5 to 10 years figuring out what the long-term toxicity is, I predict.
So Professor, next question is from Savarna. Why does it take a long time for BO thal patients to become transfusion independent? Is it something associated with the bone marrow niche or HSPC engraftment? In the same breath, Professor, when we know that sickle cell, we have a genomic, we have a prenatal diagnosis. Has anybody thought over of editing the gene in a fetal stage, or it is just hypothesis till now?
Hi, Sarana. So yeah, I think what you're referring to are the HGB-204 data. So that was, I didn't make that clear in my talk. I'm sorry about that. So the first study used the initial manufacturing procedure, and the phase 3 studies, the later studies, used this enhanced manufacturing whereby the transduction efficiency increased to 70 or 80%. So in that first group of patients where it took so long for some of the beta zero patients to become transfusion independent, their transduction efficiency was only about 30%. So I think that's the reason why. It's probably not so much. And if you think about the threshold to transfusion independence being around 5 to 6 grams per deciliter for the beta plus beta zero, and then having to be more like 8 or 9 grams per deciliter in the beta zero, again, it just takes longer to get to that plateau, particularly in the early phases studies. So I think that's the explanation, not so much the biology.
Perfect. So another question from Savarna, are the adverse effects reported after gene editing is very similar to lentivirus therapy?
Yeah, so far it looks pretty similar. Yes.
So Mr. Subroto Rai, how can just 20% correction be sufficient for cure? Is it experimental or is there an explanation?
Oh yeah, so it's a great story. It's so it's been observed in both thalassemia and sickle cell disease. And in fact, what you can do is take marrow and isolate progressively differentiated committed erythroid progenitors. And in each phase from from leading to orthochromatic and then nucleated red cells, this progressive enrichment of the donor cells. So because they're not prone to ineffective erythropoiesis, they over time naturally make up a greater proportion of those cells contributing to erythropoiesis. So, Courtney Fitzhugh and her colleagues did a pretty careful look at this after in their approach, which uses a non-myeloablative conditioning regimen, where they actually end up inducing mixed chimerism. And what they show is that at 20%, there's this inflection point around 20% chimerism where the S fraction begins to creep above 50% to the 60% range, and that's where they begin to see clinical events. So, so I think there's been some nice work over the decades, a lot of it done in Italy by Guido Lucarelli's team, Marco Andriani in particular, showing this phenomenon. So it really does occur. It's not experimental.
Perfect. Professor, 2 more questions before we wind up. Can we use some other agent other than busulfan for myeloablation, because that might be causing a problem? So we fairly remember a year back, we did a sickle cell transplant, sibling transplant, and 8 months down the line, she developed a 7q abnormality in AML. So this phenomena of lentivirus thing is not very unknown in a sibling transplant within 6 months to 8 months in sickle cell. So there's something which is happening in that particular subset.
Yeah, no, I agree. So there is another gene therapy trial that uses melphalan, but that also is a alkylating agent. You might wonder if treosulfan might be safer. I know you have access to that in India. That might be interesting to look at. But I think the field in general, and this isn't a surprise, is that would like to go into the antibody-based c-Kit antibody CD47 approach that might avoid chemotherapy altogether. Now, that's being tested in primary immune deficiency, where the benchmark is quite a bit lower. You don't need high levels of engraftment in those disorders. You need T cell engraftment, donor T cell engraftment. So or modified cell engraftment. And so that would make more sense than in thalassemia, where the bar would be quite a bit higher because of the proliferative state of the marrow. But ideally, that's what we would do. We'd use antibody immune-based conditioning regimens instead of or what we're really curious and like to investigate at Berkeley, UC Berkeley with UCSF, is to use nanoparticles to target hematopoietic stem cells in vivo. So you could give the mobilizing agent, let's say, get the stem cells in the circulation with the nanoparticles that have the gene editing reagents in them, deliver that payload, if you will, edit the cells in vivo, and then once they're in the bloodstream, they'll track back to the bone marrow and begin to contribute via hematopoietic recovery. So that might also be an interesting way to overcome this problem with chemotherapy.
Professor, very well illustrated. Thanks a lot for your time. It was really thought-provoking. Up north, you're most invited next time you're in Delhi or in India. After Corona gets subsided and you get 2 shots of vaccine. I hope you got your vaccine.
I have my vaccine, so I just need a ticket and then I'll be back.
Perfect. So Shiva is doing a great job in gene, I would say CRISPR-Cas9 technology. Let's see where it takes us to. But it was wonderful listening to you. I've been tweeting your slides without your permission, apologizing for that, for the Indian audiences to see for a wonderful talk what you have presented. So thank you, Professor. Sure, in future we will somehow connect and try to take the science to the next level. Over to Shiv. Thank you.
Thank you so much, Dr. Professor Walters and Dr. Rahul Bhargava. Thank you so much for both of you. It was wonderful listening to you. Thank you so much.
Thanks for the invitation. It was a privilege to participate.
Thank you. Bye-bye.
See you. Bye.